Look at your face in a mirror right now. Not to check how you look, to measure it. The width of your face at the cheekbones relative to the distance from your hairline to your chin. Whether your jaw is wide and square or narrow and pointed. Whether your brow ridge protrudes forward or sits flat. Whether your face is broad and short or long and narrow. Whether your cheekbones are prominent or recessed. Whether your chin projects forward or recedes. What you are looking at is not cosmetic. According to researchers at the University of Vienna, the University of St. Andrews, and the Laboratory of Human Evolutionary Studies at Harvard, the shape of your face is one of the most information-dense biological structures visible to the human eye. It is shaped by your sex hormones during puberty, by your prenatal testosterone and estrogen exposure during fetal development, by the same genetic variants that shaped your ancestors for hundreds of thousands of years, and in documented cases by specific hormonal conditions whose signatures are written into facial geometry with enough precision that researchers can identify them from measurements alone. Your doctor has probably never looked at your face as an endocrine diagnostic tool, but researchers in evolutionary biology, forensic anthropology, and endocrinology have been documenting facial morphology for decades as a window into hormonal history, ancestry, and in some cases, ongoing physiological state. The face you were born with and the face that developed through your adolescence carries biological information that most people will never be told exists. Today we are going through that information, feature by feature, because some of what is written in your face right now has been there since before you were born. Before we go through specific features, you need to understand why the face carries hormonal and genetic information at all. Because the developmental biology is what makes every finding credible rather than speculative. The face develops through an extraordinarily precise genetic program beginning in the 3rd week of embryonic development. Neural crest cells, a migratory cell population unique to vertebrates, travel from the neural tube to the face, differentiating into the bone, cartilage, muscle, and connective tissue of the entire facial structure. This migration is regulated by dozens of genes, including RUNX2, PAX3, BMP4, and the WNT signaling pathway, which also regulates skeletal development throughout the body. During fetal development, the face is exposed to the same sex hormone environment as the rest of the body. Testosterone and estrogen, present in the fetal circulation from sources including the mother's blood, the placenta, and the fetal gonads themselves, exert organizational effects on developing tissue, permanently altering the trajectory of growth in ways that are encoded in the final adult structure. The ratio of testosterone to estrogen during specific developmental windows determines aspects of facial geometry that remain fixed for life. Then, during puberty, the face undergoes a second hormonal sculpting. Testosterone drives growth of the jaw, brow ridge, and cheekbones. Estrogen maintains facial fat distribution, influences the softness of facial contours, and affects the final ratio of facial width to height. The adult face is the product of 2 separate hormonal exposures, one prenatal, one pubertal, superimposed on a genetic template inherited from thousands of generations of ancestors. This is why the face carries so much information. It is a record of your hormonal history, written in bone and cartilage and fat, visible to anyone who knows how to read it. Facial width to height ratio is the single most studied facial metric in the entire field of physiognomic research. And what the research has found about it is specific enough to be genuinely surprising. The facial width to height ratio, abbreviated FWHR, is calculated by dividing the bizygomatic width, the distance between the outer edges of the cheekbones, by the upper facial height measured from the upper lip to the brow. A wide, short face produces a high FWHR. A narrow, tall face produces a low FWHR. This ratio is sexually dimorphic, meaning it differs systematically between males and females, and the difference is driven by testosterone. During puberty, testosterone drives greater lateral growth of the zygomatic arch and mandible in males, producing a higher average FWHR. A 2008 study published in the Proceedings of the Royal Society B found that FWHR in men was significantly correlated with testosterone levels measured from saliva, with higher FWHR associated with higher circulating testosterone. The face was, quite literally, reflecting the hormonal environment that shaped it. The behavioral correlates of FWHR have been documented across multiple cultures and multiple research groups. A 2009 study in Psychological Science found that men with higher FWHR showed higher rates of aggressive behavior in controlled laboratory paradigms. A 2010 study in the same journal found that NHL players with higher FWHR accumulated more penalty minutes per game, a proxy for aggressive play, across multiple seasons. A 2011 study in the Leadership Quarterly found that CEOs of Fortune 500 companies had significantly higher average FWHR than the general male population, associating the ratio with dominance-seeking behavior and competitive drive. The mechanism proposed across these studies is consistent. Prenatal and pubertal testosterone exposure simultaneously shapes the face and organizes neural circuits in the amygdala and prefrontal cortex that regulate aggression, dominance behavior, and competitive motivation. The face and the behavior share a common hormonal cause. The face is not causing the behavior. Both are downstream products of the same androgenic environment. Look at your face. If you are male, is your face wide and short relative to its height, or narrow and long? If you are female, FWHR varies less dramatically with testosterone, but still carries information. Women with higher FWHR have been documented to show higher cortisol reactivity to stress and higher competitive motivation in multiple studies, suggesting that prenatal testosterone exposure leaves a visible facial signature in both sexes. The jaw, its width, its angularity, its projection, is the facial feature most directly shaped by pubertal testosterone in males and most directly reflecting estrogen and progesterone balance in females. And the information it carries extends beyond hormonal history into ongoing endocrine state. In males, jaw development during puberty is one of the most testosterone-sensitive processes in the entire body. The mandible expresses high densities of androgen receptors, making it exquisitely responsive to testosterone surges during adolescent development. Males with high pubertal testosterone develop wide, square, projecting jaws with prominent gonial angles, the corners of the jaw. Males with lower pubertal testosterone develop narrower, less projecting, more pointed jaw structures. This is not a continuous spectrum with infinite gradations. It is a documented biological response with measurable variation that researchers can quantify from facial photographs. A 2013 study published in Evolution in Human Behavior found that jaw width in adult males was significantly correlated with testosterone measured from morning saliva samples, with the correlation holding after controlling for age, body mass index, and ethnicity. The jaw you carry today is a record of the testosterone environment you were exposed to during puberty, written permanently in bone. In females, jaw morphology carries different information. Polycystic ovary syndrome, the most common endocrine disorder in women of reproductive age, affecting approximately 10% of the female population, is characterized by elevated androgen levels, excess testosterone, and DHEA, produced by the ovaries and adrenal glands. PCOS has documented associations with increased facial masculinization, including wider jaw, increased FWHR, and reduced facial femininity scores on standardized assessments. A 2019 study in psychoneuroendocrinology found that women with PCOS showed significantly higher FWHR and wider mandibular widths compared to age and BMI-matched controls without PCOS. This is not a diagnostic tool, but it is a documented biological association. If you are female, have a notably square jaw, and also experience irregular menstrual cycles, unexplained weight gain, facial hair, or acne, the facial morphology is one of several converging signals that warrants hormonal evaluation. Not because the jaw proves anything, but because when multiple signals converge, they are rarely coincidental. The brow ridge, the bony prominence above the eyes, is among the most ancestrally informative features of the human face, carrying information about both hormonal history and deep genetic ancestry. The continuous brow ridge of archaic humans, including Neanderthals, was one of the most prominent features distinguishing earlier species from anatomically modern Homo sapiens. Modern humans underwent facial gracilization, a reduction in facial robusticity, including brow ridge reduction, that accelerated approximately 200,000 years ago. A 2018 study in Nature, Ecology, and Evolution proposed this was driven by social selection. A mobile brow capable of expressing subtle emotions like recognition and sympathy offered social advantages over a fixed, prominent ridge incapable of such nuance. The variation remaining in modern brow ridge prominence is partially heritable and partially hormonal. Testosterone increases brow ridge prominence. Individuals and populations with higher average testosterone exposure show more pronounced ridges. A 2017 study in the American Journal of Physical Anthropology found significant correlations between brow ridge prominence and testosterone-sensitive cranial dimensions across 1,400 individuals from diverse populations. Testosterone increases brow ridge prominence. Individuals and populations with higher average testosterone exposure show more pronounced ridges. Facial symmetry is the feature that evolutionary biology of human mate preference has studied most intensively, and what the research has found moves well beyond intuitive appeal into specific biological mechanism. Bilateral symmetry, the degree to which left and right halves of your face mirror each other, measures developmental stability. In a genetically and physiologically stable developmental environment, bilateral symmetry encoded in the genome is faithfully executed on both sides simultaneously. Environmental stressors, pathogen load, nutritional insufficiency, hormonal disruption, genetic mutations introduce asymmetries by disrupting precise bilateral execution of the developmental program. Facial asymmetry is therefore a visible record of developmental instability. A 1999 study in the Proceedings of the Royal Society B found that facial symmetry was associated with immune function, genetic heterozygosity, and health history in a large cohort. More symmetrical individuals showed lower parasite load, higher immune competence, and fewer reported childhood illnesses. A 2001 follow-up found that facial symmetry assessed from photographs predicted self-reported health status with statistical significance in both sexes. The evolutionary interpretation is that facial symmetry became attractive because it was a reliable, visible marker of developmental quality, genetic integrity, immune competence, and developmental stability. Your face's symmetry, or asymmetry, is a record of your developmental history in the same way that fingerprint asymmetry, which we discussed in our previous video, records the stability of your prenatal environment. Here is what I want you to carry out of this video. The face looking back at you in the mirror is not just an appearance. It is a developmental record, shaped by prenatal hormone exposure during fetal development, by pubertal testosterone and estrogen surges during adolescence, by the genetic template inherited from ancestors, and by the developmental stability or instability of the environment in which your face formed. Look at your face again. Not to judge it, to read it, because it has been carrying this information your entire life, and now you have a framework for what it means. What do you notice about your face after watching this? What features stood out most? Drop it in the comments. I read every single one. Subscribe to the channel because next week we are going into another part of your visible biology that carries ancestral and hormonal information most people have never been taught to decode. The signals are there, and I will see you in the next video.